Welcome back, everybody. I feel like I'm losing a lot of my life trying to get this video to work properly. I clearly should have gone back and looked at the rest of these, but hey, we're going to try it again. So three's the charm, hopefully. So we're back to talk about reproduction, and in this case, we're focusing on animal reproduction. So first we want to talk about the different modes of, or at least briefly cover, different modes of asexual reproduction in animals. And there's three basic ways, if we're going to simplify this, there's three basic ways that animals can reproduce asexually. The first is by what's called budding. So in the case of budding, what's happening is literally through mitotic division of cells, a very small mini you is being developed and then grows out of your body. And that's what you see happening here with this hydra. So here's the adult mature hydra, and this is a bud. So an offspring that's developing off the side of its body. Generally speaking, with budding, the offspring is going to be literally a mini you. So it's going to be a very small version of you, which will then grow and develop into a mature individual. There's a number of different animal taxa that will do this. So jellyfish will do it, sea anemones will do it. Some flatworms will do it in this way. The second form is fission. So this is literally what it sounds like. So it's an individual that's basically dividing into two. When this happens, you have two offspring individuals that are effectively the same size as the original mature parent individual. Normally in animals, we talk about fission being binary fission, meaning they're basically dividing from one end to the other, and it's usually from like anterior to posterior. But there are other forms of vision, so it can be bilateral, it can be a number of different ways. And so the most famous taxa that do this in the animal kingdom are flatworms, but there are other organisms that will actually engage in fission. And then the third and debatably most interesting form of asexual reproduction is what's called parthenogenesis. Now, strictly speaking, this is defined as being all female individuals that produce offspring without the inclusion of the male genetic contribution. So there's a number of ways to go about this. We're not going to go into all the gory details. Offspring can be produced either via mitosis or meiosis, or by the union of products of meiosis. And some famous examples in the animal world are, of course, bees. And one of my favorites are the whiptail lizards, one species of which you see right here. But there are snakes and fishes and all kinds of invertebrate taxa that will reproduce via parthenogenesis. The way that occurs, though, differs among the taxa. And so you may find that there's some species where they don't need males at all. They can just, through environmental cues, they can usually begin the reproduction process, and their eggs will begin to divide and develop into offspring that are then, born, that are then will grow up to mature individuals and reproduce on their own. But there are some species where they actually do engage in courtship, and they might actually go through, they might actually mate with males, but then they don't actually incorporate the genetic complement into the egg. Sometimes all that's required is the sperm to sort of to initiate fertilization with an egg, and that basically stimulates the egg to divide, but then it kicks out the male sperm and doesn't incorporate it into the egg. Okay, so budding, fission, and parthenogenesis. So now I want to drill down into a little bit more detail involving the development of sperm and eggs, and then the union of those. So gametogenesis is the process where gametes are produced, and gametes are either eggs or sperm. Eggs are developed in the ovaries of females, and so that's referred to as oogenesis when those eggs are developing. And then spermatogenesis is the process for forming sperm, which occurs in testes. Hopefully you'll remember meiosis and mitosis from previous classes, but just to briefly review, If you're looking at the male aspect first, you begin with the spermatogonium, which is a diploid cell. These cells are going to be found in the testes. They'll go first through mitosis to form the primary spermatocyte, which is also by definition diploid. The primary spermatocytes will then undergo meiosis 1 and form the secondary spermatocytes, which are haploid. Those cells will undergo a second process of meiosis, so meiosis 2. They will form 4 haploid spermatids, and then those cells will go through a process to develop into mature sperm, which are then capable of fertilizing eggs. So you start with 1 diploid cell, you end up with 4 haploid cells that then develop into sperm. If we look at a little bit more detail on the anatomy of sperm, there are several different segments that you see here. So the head, which has the DNA from the male and then covered by the acrosome. This is followed by the neck, and then sort of the mid piece that is consists of an enormous number of mitochondria, which will power the sperm towards it on its journey to fertilization. And then, of course, the tail indicated here in yellow. So at the head of the sperm is the acrosome. So the acrosome contains enzymes that are capable of penetrating the egg, which is normally covered with a jelly coating to protect it. And so these enzymes will get released. Basically, it'll dissolve the jelly on the surrounding the egg and will allow it to penetrate and then release the DNA from the sperm into the egg to then unite with the maternal DNA. So the mitochondria produce ATP to power sperm movement. The flagellum is composed of microtubules which whip back and forth for swimming. And then, of course, the streamlined form is key for making it through this environment and being able to rapidly swim towards the egg. Now, as you know, human sperm are believed to last, up to 48 hours within the female reproductive tract. But in some taxa, like in some lizards, sperm will line up in this reproductive tract and they'll last for months until they're actually used to fertilize an egg. So oogenesis is similar and different from spermatogenesis. So it begins similarly. You have an oogonium, which is diploid. It's going to undergo mitosis to then develop into a primary oocyte, which is also diploid. The primary oocyte is going to undergo meiosis 1 to form 2 cells that are going to be haploid, one of which is the secondary oocyte, the other of which is a polar body, which is not going to be used in the reproductive process. The secondary oocytes are going to go through meiosis 2, which will produce 4 cells that are haploid, one of which is the ootid, the other 3 are polar bodies. The ootid is going to go on to develop into a mature ovum, and that is what's going to get fertilized by the sperm. So here's a diagram showing a sort of stylized diagram showing a human egg, and it's got sperm all around it. Important to note are these different structures of the egg. So you have the corona radiata. In blue here is the zona pellucida. Within that is the plasma membrane, and of course inside here is the cytoplasm. So these yellow bits are the yolk granules that will help to provide nutrient for the egg. And then you also have a single polar body, and then this is meant to be representing the DNA here. So this is going to carry the genetic material and nutrients to help the egg to survive, and subsequently to help give nutrients for the beginning stages of development. So there's fibrous proteins that form this vitelline envelope that surrounds the nucleus and protects the eggs. And in mammals, that vitelline envelope is referred to as the zona pellucida. So it's actually quite a challenge for sperm to be able to get through that vitelline envelope to fertilize the egg. And at the same time, there's so many sperm that are trying to do it simultaneously, but that could result in a disastrous impact if you consider that if multiple sperm were to get in there, there'd be multiple copies of DNA that would be introduced, and that could result in some mutations and failed development if that were to occur. So there needs to be a way to allow one sperm in while preventing other sperm from entering. So before we talk about that, let's first compare and contrast external and internal fertilization. So both occur within the animal kingdom. So external fertilization is normally happening in an aquatic environment, and it can be freshwater or marine. And what's happening is you have organisms that are broadcast spawning. So they're releasing their eggs and their sperm simultaneously into the water column. And then those eggs and sperm combine, unite, and then form the zygotes for whatever taxa they are. And there's a number of different organisms that'll do this. So you're seeing a starfish here. All kinds of echinoderms will do it. Sponges will do it. Clearly fish will do it. Lots of marine organisms, I dare say almost all of them, will engage in external fertilization, just as you're seeing here. Now, internal fertilization, which can happen in a number of ways, is defined, as you might imagine. So basically, the sperm are going to enter the female reproductive tract and fertilize the egg within the mother. Now, that said, you can do that in a couple of different ways. You can have copulation, as you see here with these beetles, where the male has some sort of intromittent organ or copulatory organ that he inserts into the female in order to transfer the sperm to her. But there's an alternative that you frequently see in the salamander world where the female is seen here straddling the male's tail, and he's engaging in courtship and stimulating her by waggling his tail underneath her chin. But what he's going to do is he's going to release what's called a spermatophore. So it's basically a packet of sperm that's sitting on top of kind of a proteinaceous pedestal. And he's going to lead the female over it, and she's going to pick it up in her cloaca so that the sperm pack can then open up and go and fertilize her eggs in her reproductive tract. So there's the spermatophore. And here she goes. So she's basically going to back up over it and grasp it. And then by virtue of doing this, taking the whole spermatophore in, sperm are then going to travel up her reproductive tract and then fertilize her eggs inside her. So despite the fact that he released that sperm pack out into the environment, she takes it up into her body, so it's still internal fertilization. Now, we're going to talk about this in lecture, but just as a highlight, so if you look at the copulatory organs throughout the animal kingdom, you will find that there's an enormous amount of variation in those structures. So some of them have support structures inside them, usually bones that are called bacula that help to keep them rigid so that they can effectively breed with individuals in their species. What I'm showing you here are images of hemipenes from snakes. So snakes and lizards all have what are called hemipeneal structures. So they're not all called penises. They're called hemipenes because instead of it being a typical sort of hose like a regular penis, it's a structure that has a groove along the outside of it, and sperm travels along that groove and then flows and then fertilizes. So they insert the entire hemipenis, the sperm flows along the groove, and then it goes into the reproductive tract of the female, and then sperm travels up and fertilizes her eggs. So not only is it not a hose, but a grooved structure. In addition, they have two hemipenes. They have one on the left side and one on the right side, and they alternate use when they're breeding with females. And so what you're seeing here is images of the hemipenes of some snakes, and they're showing you the front side and the back side. That's what these images are. So sometimes they're unilobed, as you see here. Sometimes they're covered in this colliculate material that's kind of spongy looking. Sometimes they have spines. Sometimes they've got lobes on the top and all kinds of rigidy structures on them. And sometimes they're hugely bilobed. So this whole structure right here, this is one hemipenis that gets inserted into the female for reproduction. And so this is one of the species of rattlesnakes in the southwestern United States. So rattlesnake hemipenes are just amazing. The point, though, is the big question is you see so much variation in these copulatory structures. Why would you see that? So what's the explanation for that? You would think just a tube is a tube is a tube. You don't need anything special to transfer the sperm, but that's not what we see. We see an enormous amount of variation. So why is that? So we'll discuss that on Thursday. So be thinking about it. Yeah, I'm so fascinated by these things. I actually have pictures of hemipenes on my wall, so you can probably see them behind my head. We'll see this video in class. Okay. Right. So the fifth objective involves drilling a little bit down further on that fertilization process. And this is a great image, by the way, that's showing this vitelline envelope around the egg. But let's talk about this process in a little bit more detail. So fertilization is defined as the union of egg and sperm to form that diploid zygote. But again, there needs to be some sort of process to prevent more than one sperm from getting into the egg. How does that work? So in this diagram, here's the egg. This is the genetic complement from the mother. You can see the jelly layer, and we're going to start here. So for many taxa, eggs will actually release a chemical or hormonal signal that attracts sperm to them. Once there, they're going to need to find some way to make it through this jelly layer. And on the heads of sperm is this acrosomal layer that is composed of enzymes that help to break down that jelly layer so that they can then enter the egg proper. When they do that, and you see this happening here, so here's a sperm that's then hit the jelly layer, and this is supposed to be indicating that the jelly layer is dissolving. And at the same time as they're releasing these enzymes, the flagellum is actually frantically wiggling or beating. So it produces some sort of physical force to try to push its way into the egg and penetrate. So once the sperm actually makes it to the egg, it will then fuse with the plasma membrane and release its DNA into the egg cytoplasm, which will ultimately unite with the maternal complement and then form that initial zygote. So once that sperm actually hits in here, what happens is there's going to be a series of reactions that will then prevent other sperm from entering the egg. And this is how it works. So once the sperm nucleus enters the cell, goes through the cell plasma and enters into the cytoplasm of the egg, the entire egg surface depolarizes and prevents other cells, so there are other sperm from getting in. Here's how it works. So here's an egg that's surrounded by loads and loads of sperm. And right about here is where a single sperm enters the egg. When that happens, it causes a release of calcium ions from the endoplasmic reticulum. When that happens, vesicles called cortical granules, they fuse with the plasma membrane of the egg, and then that causes them to release their contents via exocytosis. And when these enzymes are released, what it ends up happening is it basically blocks the receptors for sperm on the surface of the egg, and it effectively just vacuums away all of those receptors, and so it just pushes all the sperm from the surface of the egg and prevents any more of them from being able to enter into the cytoplasm. So here you see the beginning of what's called the fertilization envelope to form here. That's basically going to wipe off all the sperm. And here it's basically completed formation. So it's a really cool process. Okay. So other concepts that you need to be familiar with animals and animal reproduction is viviparity, oviparity, and ovoviviparity. So most mammals are viviparous, and sometimes this is loosely defined as live birth. But to be more specific about it, what it means is for the offspring to develop completely within one of the parents, and in this case it's usually the female parent. And not only is development completely within the mother, but when the offspring are born, They are not surrounded by any sort of covering. So there's no egg involved. You just have the offspring being born from the mother live. Now I say most mammals, so you'll recall, I hope, that there's three major branches of mammals. You have the placentals, which you are an example of. You have marsupials, which are the pouched mammals, so things like opossum and kangaroos and koalas. So the offspring also develops. Most of the development is within the female, but it's not always within the uterus. And then you have the monotremes. So these are things like duck-billed platypus and echidnas and things like that. They lay eggs. And so in that case, development is not within the body of the female. They lay those eggs in nests and then care for them. And actually, it's more complicated than that. But anyway, development is not within the female. Now, normally we think of birds as being examples of oviparity. So they, like echidnas and duck-billed platypi, so they have offspring that are completely enclosed within eggs. And here it says it's protected by a hard shell, hard-shelled eggs. But actually, if you look at things like pretty much all the reptiles that lay eggs, those eggs are leathery, but they're not really hard. Like a chicken's egg. So they're pretty leathery, but they still protect the egg, the embryo, nevertheless. So in this case, you're talking about the development of the offspring in an egg outside of the parent. So that's oviparity. And here's some examples of little babies coming out of eggs. Then there's the adult. So ovoviviparity is a condition where you have development with completely within the female, but it's within the egg. So offspring are within eggs that are developing within the parent, and then when it's time to be born, what happens is they basically emerge from the egg within the female, and then when they come out, it looks like you see in this picture here, where it looks like normal live birth, like many mammals do. But in fact, that individual developed within an egg within the mother. That's ovoviviparity. Okay, that's all I got for you. We'll talk about more issues in class on Thursday.